Boa tarde, pessoal. Então, a gente está aqui para mais uma Conferência Banting. E hoje a gente vai receber a professora Daniela Drummond-Barbosa. Ela é brasileira e americana. E ela fez biologia na UFMG. E aí ela depois foi fez o doutorado dela em Yale, trabalhando com virologia. E depois ela fez o pós-doc dela mudando totalmente de área, estava conversando aqui antes, mudando totalmente de área para Drosophila, e hoje ela é professora na Hopkins University, lá nos Estados Unidos, e tem trabalhado usando Drosophila como modelo para entender respostas das células-tronco, principalmente células-tronco de ovário, no caso, como modelo, mas como que as células-tronco respondem ao ambiente de modo geral, então nutrição e outros fatores. Eu tenho certeza que vai ser uma apresentação muito legal. Professora, muito obrigado por ter aceitado o convite. Então, deixa eu dar alguns recados, que eu sempre esqueço desses recados antes. Vocês podem, como sempre, vocês podem botar as perguntas no chat, tá? Que aí a professora Daniela vai responder no final. A professora Daniela vai fazer a apresentação dela em inglês, porque tem pessoas do laboratório dela, pós-docs e alunos de doutorado lá, que não são brasileiros, que vão assistir também, então ela vai falar em inglês para poder todo mundo poder acompanhar. E eu vou botar a lista de presença no chat para vocês responderem daqui a pouco. E eu acho que todos esses são esses recados. Professora, muito obrigado por ter sido o convite, acho que vai ser sensacional. Fica à vontade, a tela é sua.
Muito obrigada pelo convite, Davi. Mas agora eu vou mudar para o inglês, porque meu português científico é muito ruim e tem pessoal do meu laboratório aqui também. So the broad question that my lab is interested in is, how does oogenesis respond to the external environment and physiological factors? And here you can see a lot of different types of ovaries or the worm gonad. And despite all the very different morphology, what they all have in common is that they are producing eggs, and that they have to be highly tuned to the environment so that they adjust the production of eggs to the conditions around them. And our favorite ovary to study are the ovaries from fruit flies. And so I'm going to start by giving a brief introduction of the Drosophila ovary. You guys might already have heard this before, but I'll give my own introduction just so that we are all on the same page. So each of the Drosophila ovaries has 16 to 20 ovarioles. And what you can see here is one of these ovarioles. And the anterior portion is called the germarium. And then the germarium is followed by progressively more developed egg chambers or follicles that support the development of the oocyte. Each follicle has a 16-cell cyst in the middle, and that cyst is surrounded by follicle cells. And one of the 16 cells in the germline cyst becomes the oocyte, and the other 15 cells are nurse cells that will support the growth and development of the oocyte. And all these follicles are formed in this anterior germarium, and you can see a bigger version here. And there are both germline stem cells and follicle stem cells that give rise to the germline and somatic components of the follicle. And the germline stem cells are present in a specialized niche composed of cap cells, and each time they divide, they give rise to a cystoblast, which then goes on to divide to form 2, then 4, then 8, then 16 cell cysts through synchronous mitotic divisions. And these cells all remain interconnected. And this is how whatever the nurse cells produce can be transported into the oocyte. So one reason why I was very interested in focusing on the Drosophila ovary to understand, in particularly, the response to diet. When I joined Alan Spreading's lab in the late 90s, was because the ovary has a very drastic response to diet. And it was known for a long time that the 2 points of regulation of oogenesis were at the onset of vitellogenesis or yolk uptake, which happens in stage 8 in Drosophila. There's 14 total stages. Vitellogenesis happens during stage 8, and a mature stage 14 is ovulated to be then fertilized and laid as an egg. And in addition, other than these 2 points of regulation that were known, there was nothing else known about earlier stages of oogenesis. So when I was a postdoc, I used a combination of approaches, including a lineage tracing approach, and I showed that there were differences in germline stem cell proliferation, so they divide faster when the flies have a lot of yeast on their food and slower when there's no yeast. We also found later in my lab that the maintenance of the germline stem cells in the niche is also regulated by diet, and on a yeast-free diet, stem cells are lost faster. And I also found that the division and growth of the germ cells and of the follicle in general, including the follicle cells surrounding the germ cells, is regulated by diet. So everything happening faster on a yeast-rich diet and slower on a yeast-free diet. And then on a yeast-free diet, you also get a block in vitellogenesis, and the mature oocytes are retained in the ovary instead of getting ovulated. And as a result of all of these processes, the females will lay many more eggs on a rich diet than on a poor diet. So the question that I've been asking ever since I started my lab in 2002 what are the physiological mechanisms mediating this response to diet? So, and I'll give you some brief examples of what we found along the years, starting with the very early work from my lab in the early 2000s, showing that insulin-like peptides that come from the brain act directly on the ovary to control the number of stem cells maintained in the niche, to control how fast they divide to make new cystoblasts, to control follicle growth, and to control vitellogenesis to help mediate the response to diet. And the reason why I focused first on insulin signaling is because of the obvious connection in many organisms between diet and insulin signaling. And I'll just show you one example of how we found this direct link between these insulin-like peptides and the ovary. And a lot of the work we did was taking advantage of genetic mosaic analysis. And I will not go into the details of the genetic tools we use for that because of time limitations, but the important thing is that we start out with an organism where all cells have wild type copy of the gene of interest. And then we can control at a specific time some mitotic recombination events that will eventually lead to some cells becoming homozygous mutant for the gene of interest. So you start with a heterozygous animal that has a wild type and a mutant copy of a gene, and then you end up with a heterozygous animal that has a few cells that became homozygous mutant. And you can recognize the homozygous mutant cells because they lose a marker that's closely associated with a wild type gene. So in using the system, for example, we showed that the insulin-like peptides signal directly to the germline to control follicle growth. So in this case here, it's a control ovarial. So you can see some red cells and some non-red germline cells. In the case of the control, they are all wild type cells. And you can see the normal growth of the follicle, and all the follicles stay in order of growth as the same order that they are formed in the germarium. But if you generate homozygous mutant clones, which are recognized here by the lack of red in the germline here, using two different alleles of the insulin receptor, you can see that these insulin receptor mutant germline cysts have very delayed growth compared to their neighboring wild types. So this one is older than this anterior one here, but it's smaller. Same thing with this one relative to this one. So this shows you that if you don't have the insulin receptor in the germline, there's a problem with follicle growth. So that tells you that insulin-like peptides then have to signal directly to the germline through that receptor to control their growth. And on using a very similar approach, just switching here from red to green, we show that if we mutate instead the target of rapamycin gene in Drosophila using a strong loss of function allele, you also see very delayed growth. So this egg chamber is younger and should be less developed than these two, but these two are grossly delayed because they don't have the TOR gene in the germline. And conversely, if you instead mutate the negative regulator of TOR, TSC1, then you have overactivity of TOR, and the oocyte and the germline cyst grows and the follicle grows too much or too fast. So so that just gives you a flavor for how we figured out this connection here. And using very similar approach, we show that there are other diet-dependent signaling pathways that are intrinsically required in the ovary. For example, I already mentioned TOR, and in the case of the follicle growth, TOR and insulin are working together. But TOR has some insulin-independent roles too in controlling the response to diet. We also found roles for AMP kinase, steroid hormones, and the adiponectin receptor, which are all connected to diet in one form or another, but I won't go into the details there. So at the next phase of my lab, we became interested in more than what's signaling pathway has to be activated directly in the ovary, but more broadly in terms of what other tissues might be receiving in terms of dietary signals, which they would then communicate via secondary signals to the ovary. So, because in reality, as the organisms eat, all different organs can sense that what they ate, and different organs might react differently to that diet. And in order to coordinate the response of the organism as a whole, they have to constantly communicate with each other to finalize, fine-tune how any given organ responds to diet, including the ovary. But of course, we can't study all the arrows here at the same time. So we focused on this connection going from adipocytes to the ovary, and we asked, can adipocytes communicate the nutritional status of the organism to the ovary? And the reason for focusing on the adipocytes at first is because both in mammals here, adipocytes have an endocrine role. So the adipose tissue is nutrient sensitive. Adipocytes produce various hormones and other systemic signals. And in agreement with this important role in this endocrine role, when, for example, when women have either severe obesity or they lack fat cells, there is a link to higher risk of infertility. So, and similarly, you guys are very familiar with insects and other arthropods, but the Drosophila adipocytes are part of an organ called the fat body. So, and the cell types are adipocytes and inocytes. And the adipocytes have similar roles to mammalian adipocytes. The inocytes have similar roles to liver cells, and this fat body also has an immune role, as you guys know. So, and the evidence, for example, of the fat body being an endocrine organ, for example, during larval growth, insulin and TOR signaling from the larval fat, larval fat can indirectly control organismal growth. And both in the larva and the adult fat, there are factors sent to the brain to control insulin secretion. So we asked the question, does amino acid sensing in the adipocytes get somehow communicated to the ovaries? So the idea was to leave the entire fly pretty much intact so that all organs, including the ovary, would be able to sense directly the presence of amino acids. But we only disturbed the amino acid sensing process in the adipocytes. So we use the UAS GAL4 system, which I will not go into details on today, but basically we used a very specific promoter that only drives in adipocytes to drive RNAi against individual amino acid transporters. So flies, Drosophila has about 40 different amino acid transporters that are either predicted or shown to be amino acid transporters. And we knock down one by one of all the ones available that had RNAi lines available, specifically in the fat cells, in the adipocytes. And then we asked whether or not we saw any changes in oogenesis. So this, just to make sure, these are very, very small manipulations in amino acid sensing, because we're just knocking down one amino acid transporter at a time, and the knockdown was not very efficient. So it will be the equivalent of having a small dip in the levels of amino acids going to adipocytes. And what we saw was that when we got rid again of these amino acid transporters in the adipocytes, one by one, but just a small decrease, we saw when we counted the number of eggs laid per female per day, that if we looked at day 12 after the RNAi was started in the adult females, you could see that there was a significant decrease in egg production. And I'm just showing you the examples for 3 different amino acid transporter knockdown. So what steps of oogenesis then are being affected to reduce egg production when just the adipocytes sense less amino acids? So I can't show all the data, but I'll just summarize here that we showed that the amino acids controlled, amino acid sensing in the adipocytes controlled the number of germline stem cells in the niche of the ovarioles and controls ovulation. So that when you knock down the amino acid transport in adipocytes, there is a reduction in the number of germline stem cells and a block in ovulation. So the stage for mature eggs accumulate in the ovary. And we showed that target of rapamycin is required inside the adipocytes to mediate these effects of amino acid sensing on ovulation. So if we directly block TOR signaling, we get a very severe accumulation of eggs in the ovary. But to our surprise, TOR had nothing to do with the control of germline stem cell numbers. So we were interested in knowing what is the signaling mechanism downstream of amino acids that's controlling the stem cell numbers in the adipocytes. So the next logical candidate, since Tor wasn't it, was the amino acid response pathway, which was originally identified in yeast. And it's basically a pathway that gets activated when there's one or more essential amino acids missing. In the case of yeast, is to induce the expression of biosynthetic enzymes. In the case of flies and mammals, this pathway has remained conserved, but instead, many times it functions as a stress response pathway. So normally, this pathway is activated when there is not enough amino acids of one type or another, so that when the amino acids are coupled to tRNAs by aminoacyl tRNA synthesis, not all the tRNAs will be coupled to an amino acid because there's not enough amino acids, and that uncharged tRNA will bind and activate the GCN2 kinase, which will then phosphorylate EIF2 alpha to inhibit global translation, but to induce expression, transcription of a select number of targets through the function of ATF4. So we wondered if this pathway might be responsible for the reduction in stem cell number in response to amino acid transporter knockdown in the adipocytes. So one prediction, if that was the case, is that even in the normally this pathway works like I mentioned, right? If there's normal amino acid levels, all the tRNAs are coupled, the pathway is off, and that would be a normal number of germline stem cells. That's our model, right? And if there was reduced amino acid levels, there would be more uncoupled tRNAs, and the pathway would be on, and it would reduce the number of germline stem cells. So the prediction was that if we had normal levels of amino acids, but we genetically, by doing RNAi against individual aminoacyl tRNA synthetases, prevented coupling of the tRNAs, then we would expect a decrease in germline stem cell numbers because this would experimentally activate the pathway even when there is normal levels of amino acids. And I won't be able to show you the data, but the answer was yes. When we prevented coupling of transporters of tRNAs to the amino acids, we got a reduction in stem cell numbers. But I'll show you the results from the second prediction. The second prediction would be, okay, normally if we knock down the amino acid transport in the adipocytes, you wouldn't have enough of a certain amino acids in the cells. So that would create the extra tRNAs and would lead to decrease in the number of germline stem cells because the pathway got activated. So if we still had the same exact situation, but we knocked down GCN2, we should be able to suppress the decreased number of stem cells and get normal numbers of germline stem cells. And these are the results that we got. So I'll just walk you through the controls. This is the number of stem cells shown here. There's usually normally like around 2 or 3. In this case here, you can see in the control as 0 days in black here. You can see on average 2.5, meaning 2 or 3. The germline have 2 or 3 germline stem cells. And there's a normal rate of stem cell loss with age that we see here. So at 10 days, that number went down. If we knock down just the amino acid transporter to decrease the amino acid levels a little inside the adipocytes, the germline stem cell number in each ovary goes down faster. But if we combine this knockdown together with the knockdown of GCN2, that kinase that's required to activate the amino acid response pathway, then we get suppression of the stem cell loss, and you see very similar rates of loss of stem cells if you look at the slopes of the curve as the control. So then, to summarize a lot of work into this one slide, then adding on to what we had shown before, this postdoc in the lab, Alyssa Armstrong, showed that amino acids then control ovulation through TOR and control germline stem cell numbers through GSEN2 when they are sensed in adipocytes. So there's clearly communication from adipocytes to the ovary regarding nutritional status. And again, I don't have time to show either, but Alyssa, the same postdoc, also showed that the story with the insulin-like peptides is not so simple, because in addition to signaling directly to the ovary, insulin-like peptides also will signal to the adipocytes, and the adipocytes will in turn communicate the status of insulin in the body and further refines the response of the ovary to diet. So, and we continued our interest on adipocytes by asking now a slightly different question, right? Because we were focused on adipocytes receiving certain stimuli here. But now we were interested in understanding, like, how do adipocytes relay nutritional information to the ovary? And we didn't exactly answer this question, but that's how we started our next line of investigation, because we thought, okay, if adipocytes sense nutrients, they could be sending secreted factors either directly to the ovary or to an intermediate organ, or signaling metabolites, or they could communicate to the ovary simply by mobilizing certain nutrients. So we took a quantitative proteomics approach called iTrack, and I won't go into too much detail, but we basically hand dissected hundreds and hundreds of fat bodies from flies, either maintained on a rich diet, rich meaning with yeast, a lot of yeast, or flies who were compared to flies who were switched just for 12 hours to a yeast-free diet. And then all these dissected fat bodies were in quadruplicate, actually, for each condition, were subjected to, they were all labeled in a different way, so you could compare the relative amounts of all proteins among all 8 samples, right? The quadruplicate for rich and the quadruplicate for poor. And just a point of clarification, the reason why we chose 12 hours is because we knew that oogenesis could be downregulated in under 18 hours, 18 to 24 hours. So we knew it was a fast response to diet. So, and we wanted to catch early proteins that change in the fat. And also the 12-hour time point, there is no obvious change in the adipocytes after just 12 hours on a poor diet. So it was like ideal for identifying primary changes in response to diet. And what we found, we were able to identify a total of 2,500 proteins. This is not a very sensitive method. It only detects more abundant proteins. So, but we were able to identify 2,500, and about 450 of them were significantly either down or upregulated on one diet versus another. And this graph, this volcano plot, is just showing the p-value. So anything above this red line here has a significant p-value, and anything that's to the left of this line or to the right of this line represents a significant, represents a change in the level. So you have to focus on these portions here. So there's 224 proteins that fall into this spot, which means they are lower on a poor diet than on a rich diet, and these ones are the opposite. They are lower on, they are higher on a poor diet and lower on a rich diet. And just to give you, we saw that many of those proteins that were regulated were metabolic enzymes, but there were other categories of proteins too. But we decided to focus on the metabolic enzymes because it was such a predominant group of proteins that were being regulated. And basically all the ones that are shown in red here, and you can see this is for the metabolism of carbohydrate, energy metabolism, nucleic acid, amino acid, lipid metabolism. And you can see that everything shown here in red are enzymes that are higher on a rich diet than on a poor diet. And all the ones shown in blue here are higher on a poor diet than on a rich diet. And just to give you some examples, on a rich diet, there seems to be a shift of glucose metabolism towards the production of pyruvate and acetyl-CoA. So you can see a lot of these enzymes here are upregulated on a rich diet, but they are downregulated on a rich diet and compared to a poor diet inside the mitochondria, like the TCA cycle, the oxidative phosphorylation enzymes are downregulated compared to these. So it suggests that maybe this the products of this earlier part of the metabolism here are being used as building blocks for the ovary, or it could be that acetyl-CoA is causing epigenetic changes. We don't know. The other example, we saw there was increased fatty acid beta oxidation in the adipocytes. So that could be maybe on a rich diet for ATP production or production of ketone bodies to be transported to other organs, maybe the ovary too. And the other example here, we saw increased phosphatidylethanolamin synthesis, which could be involved in signaling or control of lipogenesis. And actually, in this paper we published from my former postdoc, Shinya Matsuoka, we did examine all three pathways. But I'm just going to give you one example today of how we tested whether these enzymes that are rapidly regulated by diet have any roles in oogenesis indirectly. So in this example, then I focused on these enzymes here, which are regulatory enzymes that are involved in pyruvate and acetyl-CoA production. And if you focus first on the right side here, we were examining specifically early germline cyst survival. So what you see here is the very tip of the ovariole where the germarium is. And you can see this is a control germarium where we knock down luciferase, an unrelated gene. And DAPI in blue is showing the nuclei. And we have some other markers to highlight other structures in the ovary here. And this CP1 in red is recognizes the cleaved caspase, this CP1. So whenever you see the red staining, there are dying germ cells. So, and you can see in the example of HEC RNAi or PeptiK RNAi, some examples of dying germ cells. And you can also notice that these germaria are very short, probably because cells have been dying there for a while. And when we quantify the percentage of germaria that have dying cysts, if you compare the control with HEC or two different PeptiK RNAi lines, there's a significant increase in the death. And we did not see an increase with these individual enzymes knocked down, but in data that's not here, when we combine the RNAi for both of them, we also saw a significant increase, suggesting that enzymes involved in production of acetyl-CoA or pyruvate are important for promoting germline cyst survival inside the germarium. And we didn't see any effect on germline stem cell maintenance or proliferation, and no effect on vitellogenesis, showing there's a very specific effect, not as a general sickness of the fly or something. So just to add on to this growing diagram here, so Shinio showed that diet-dependent metabolic pathways have roles that are very specific in regulating the steps of oogenesis that are sensitive to diet, but we don't know if it's through metabolites or through some other mechanism. So now, this last two short parts of the talk, I would like to focus on more recent work from the lab from two postdocs. The first one is Leisa Weaver, and she actually left the lab a year ago, and she's now an assistant professor at Indiana University in Bloomington. But the question she asked when she started my lab was she was interested in more roles of nuclear receptors in controlling the ovary in response to various external or physiological inputs. And in her case, her focus was particularly on nuclear receptors that are required outside of the ovary in other organs, in peripheral tissues, and play more indirect roles in oogenesis. And the reason why she focused on that is because most people tend to focus on the nuclear receptors that are required within the ovary itself. And we had done the same thing too in the past, looking at the role of the ecdysone receptor in the ovary. But we wanted like a broader view, more whole organism wide. And as you guys know, nuclear receptors control diverse physiological processes, including reproduction, metabolism, immunity, and many other processes. So, and what's interesting about nuclear receptors is that they are transcriptional factors that can regulate transcription, but at the same time, they have a ligand binding domain that makes them able to be regulated by a variety of systemic signals that are in your circulation. So they can literally connect your physiology directly to the transcriptional program of a cell. And there's previous work from my lab and other labs had shown that there are nuclear receptors mostly required in the ovary itself that influence Drosophila oogenesis. And I'm not going to go through every single one in detail, but that includes the ecdysone receptor and other nuclear receptors. And all of them in this case are required within the ovary itself, with the exception of the ecdysone receptor that has an additional role in the brain in controlling feeding behavior. But other than this example, there was not very much known about other nuclear receptors functioning in peripheral tissues. So Leisa did actually a systematic screen where she knocked down throughout the body of the flies each one of the 20 or so nuclear receptors in flies. And she selected to.
Someone needs to sign for a package. Someone, I didn't know class.
I'm in the middle of a seminar. Sorry. Sorry, I'm at home, obviously. There's some things happening. She showed that if you knock down ubiquitously throughout somatic cells, the nuclear receptor 7-up, for example, there is a significant reduction in egg production compared to the control females at 5, 10, and 15 days of RNAi in adult females. So what is 7-up? So in Drosophila, 7-up is a nuclear receptor that controls eye development and nervous system development. And in mammals, the homologs are CoopTFs that have a number of different roles, including tumorigenesis, angiogenesis, eye and nervous system development, and control of luteinizing hormone expression. So when Leslie then went to figure out what specific steps of oogenesis were affected to give rise to that reduced number of eggs, she looked closely at the germarium and to start out, and she quantified the number of stem cells over time. So this is a controlled germarium, and the germ cells are labeled in red here, and the stem cells are highlighted here, and they are right next to their niche. And what you can see is in this example of 7-Up knockdown, there's only one stem cell here, and there are fewer germ cells in general. And if you compare the control RNAi in gray to do two different 7-Ups RNAi, and you focus on the slope, which is the rate of decrease of stem cell numbers, there is definitely a higher rate of stem cell loss when 7-Up is ubiquitously knocked down. And similarly, there was also an effect on germline cyst survival. So this is a controlled germarium here, and this is a 7-Up knockdown germarium from a ubiquitously knocked down female. And in this case here, the dying cysts are labeled by tunnel labeling. So essentially, the cells that are dying have a lot of broken ends of DNA, and they incorporate these green nucleotides that will make the cells shining green here. So when she quantified that, if you just focus on two different regions of the germarium, the more anterior region and the more middle region, when you compare before the RNAi starts at 0 days with 10 days of RNAi, if you compare to the control, there's a significant increase in the percentage of germarium with dying cells relative to control. And finally, the third phenotype that she saw from ubiquitous knockdown of 7-Up was the death of vitellogenic follicles. So this is a control ovarial, just knocked down for luciferase, staying with DAPI. And you can see the nice progression of the follicle development. And this is a large vitellogenic egg chamber here with the oocyte filled with yolk that you can't see from this picture. And then this is a ovarian from a 7-Up knockdown female, and you can see this is a dying vitellogenic stage. And when she quantified that, she saw that compared to the controls, there was a significant increase in the percentage of ovarians with dying vitellogenic follicles. So to summarize a lot more work than that she had in her paper, Leslie also was able to figure out what specific tissue required and cell type required 7-Up for these effects on germline stem cell maintenance, cyst survival, and vitellogenesis. And she found that 7-Up was required in adipocytes. Again, we can't escape adipocytes. Was required in adipocytes for the maintenance of stem cells and to promote cyst survival. So when you knocked it down, the stem cells got lost faster and the cysts died more at a higher frequency. But she found that for vitellogenesis, 7-Up was actually required in the inocytes and through presumably a different target. So there's lots of questions that this work raises in what are the inputs upstream of 7-Up, because it's an orphan nuclear receptors. What are the transcriptional targets of 7-Up, whether or not the mammalian homologs have similar functions, and whether other nuclear receptors have, similarly complex roles in controlling oogenesis. And along those lines, Leslie also found a similarly complex role for another nuclear receptor called HR4, which is the homolog of GCN4 in mammals. And that one is actually required in the germline, in the niche of the germline stem cells, and in muscles for different roles in oogenesis. And that paper was published earlier this year, but I don't have time to to go into that. And also a paper we published earlier this year or last year, now I'm confused. It's another paper following up on this 7-Up story, where Leslie used RNA-seq, and she identified as the targets of 7-Up many different genes involved in immunity and xenobiotic responses in the fat body. But we don't know yet what is the functional connection between these targets of 7-Up and these responses in oogenesis. But as I mentioned, Leslie has her own lab now, and she will be pursuing these questions herself in her own lab. And now for the last but not least part, I'm going to talk about the work from Ana Caroline Gandara, which I think a lot of you know, including, the host. And Ana is now a postdoc in my lab. She's been in my lab for, I think it's like 3 years or so. And she did all her previous training at UFRJ, so you guys might be very familiar with her. But I will talk to you about the work that we just submitted yesterday. The manuscript was submitted yesterday for this work that she did, and we're very proud of this work. So she's interested in how temperature might affect oogenesis. So as we all know, in case anybody's wondering why we should be worried about temperature, there is a major problem with climate change, and it's impacting a wide range of organisms and drastically changing the environments where they live, not just in terms of temperature, but in terms of like habitat availability, etc. And I'm sure I don't need to argue for this audience how important insects are, and arthropods, but they can be, they're important for food production, both because they can be agricultural pests or because they themselves can serve as food. They represent lots of different important disease vectors, and they're also important for agriculture in other ways, like through pollination. So what Ana found and I should also mention that most of the work that had been done addressing temperature in Drosophila before was really focused on flies that developed at the suboptimal temperatures. So it's really hard to tell what's adult specific if the entire development occurred under a certain condition. Or alternatively, the studies were done in adult flies, but at very, very high temperatures where they wouldn't be able to survive for prolonged periods of times anyway. So we chose instead to focus on suboptimal temperatures, which are actually temperatures that everyone that works with Drosophila in the lab use these temperatures routinely. 18 degrees, which we're calling cold, 25 degrees, which is the control, and 29 degrees, which we're calling warm. And what Ana found was that if quantifying the number of eggs laid per female per day over time, was that at 25 degrees, of course, as the young females, after they eclose out of the pupal case, they'll take a little while to ramp up egg production. And then they'll continue to maintain higher levels of egg production, but as they get a little bit older, egg production starts going down. But she noticed that whether she maintained the flies at 18 degrees in blue here or at 29 degrees in red, there was a significant reduction in the rate of egg production compared to the control 25. So the question then that Ana asked is, what are the physiological mechanisms mediating these responses to warm and cold temperatures? Because both of them decrease, but are there similar mechanisms involved, or are there different mechanisms? What steps of oogenesis are being affected? All these were relevant to this question. So she walked through the different stages of oogenesis, and I'm just going to show you the key findings, because hopefully you'll be able to read her whole story when the paper is eventually published. So she looked, for example, at cell death of early germline cysts. As I already mentioned several times before, this is a point of regulation by physiological factors. So again, she used APOP-TAG, the same tunnel labeling technique for dying cells, and she quantified the frequency of germmeria at 18, 25, and 29 that had APOP-TAG positive germ cells inside the germarium. And when she quantified this data, both have 5 days of incubation at these various temperatures after the flies had closed to adulthood. So the development was all at ideal temperature, room temperature. So, and 15 days. And what she sees, even at the early 5-day time points, compared to the control, the warm temperature causes a significant increase in cysts that are dying inside the germarium. And to our surprise, like the cold temperature actually improves the survival of cysts, and we see even fewer dying germline cysts inside the germarium than we see in the case of the control flies, which is in this case, it would be a beneficial effect of cold, which is not really contributing to the reduced number of eggs they produce. But in this case, the increase in death of the early germline cysts could contribute to the reduction in the number of eggs produced by the flies at 29 degrees, the warm temperature. And warm temperature also increases vitellogenic follicle death. So you've already seen some pictures like this, but this is the image showing the ovarioles at 18, 25, and 29. And you can see the red arrow here is pointing to a dying vitellogenic egg chamber that's present in this ovariole instead of a normal vitellogenic egg chamber. And when Anna quantified this data, it's interesting that at 5 days and 10 days, there's no significant difference. So this is clearly a different thing from the early dying cysts. But in the case of vitellogenesis, the death occurs only at 15 and 20 days, which sounds like a separate process that gets, comes into play after longer periods of incubation at 29 degrees. So just to summarize this warm temperature effect. So basically the warm temperature will induce the death of these early germline cysts and induce the generation of vitellogenic follicles. And that's how it's contributing to a reduced number of eggs produced at 29 degrees. So what about the 18 degrees? Anna looked at all same processes at 18 and 29, but I'm just showing you the ones that matter in each temperature. So for determining, it turns out that the cold temperature reduces follicle growth rates. And the experiment that she did to show that, was a lineage tracing system that is also based on FLIP-FRT. So I'm not going to go into the details of how this lineage tracing works, but basically at time 0 of the experiment, in the intact living flies, Anna can induce the labeling of individual germline cells in this very anterior region of the germarium, where they are mitotically dividing to form the 16 cell cysts, but no other later stages are labeled for the germline at this time 0. And then what she does at later time points, well, as I mentioned, the cysts were originally labeled here, and the label is of a green color. The depth is just showing the nuclei, and this red is just highlighting the morphology of the ovary. So she knows they were labeled here. So she looks at different time points, and she can see that some of these germ cells that were labeled inside the germarium have progressed farther into oogenesis and are now part of these later egg chambers, right? So she just quantified for each ovariole what was the farthest, most developed germline cyst that had a labeled cell, because that was a measure of how far the first one that was formed a follicle was able to progress. And then she quantified this data. So this is the most developed labeled cyst, for example, was shown with the arrowheads here, the one that progressed farther for each ovarian. And then she plotted the distribution of this posterior most labeled cyst, the most developed, inside the germarium and progressing through the ovarian. So these stages here correspond to the stages along the ovarian. And you can see after 1 day at 18 degrees, most of the labeled cysts are still inside the germarium. While at 25 control and at 29 degrees, the warm temperature, they've already left the germarium. And as you look at later and later days, you can see that the labeled cysts at 18 degrees tend to be less well developed and have moved less along the ovaries than the ones at the control and 29 degrees. So this shows you that these egg chambers at 18 degrees, these follicles, are developing more slowly and growing more slowly at the lower temperature. So, and it turns out that this is the major reason why egg production is reduced at cold temperature. So you can see very different mechanisms. But of course, it's not just the number of eggs that matter when it comes to fertility and the ability to give progeny, right, of the females. Oocyte quality also matters, because after you produce a certain number of eggs and they're fertilized, the question is, are these eggs able to support development of the future embryo inside the eggshell, right? So the readout that Anna used was whether or not the eggs that these flies laid were able to hatch into larvae. So, and what she did then, she looked at the eggs laid by females after they had been 20 days at 29 degrees or 25 degree control. And to rule out that anything going on with the males could influence these results, she put young males at the very end of this incubation period together with the females, replacing any old males that might have been in there, to make sure that these females that had been there for 20 days had access to young males that are not really being incubated for that long of a time at these temperatures. So just looking at the female effects. And you can see that if the females were kept at 25 versus at 29 for 20 days, there is a drastic decrease in the number of eggs that are able to hatch into larvae at 29 degrees, showing that oocyte is really impaired. And it didn't matter if these females had old males, had males as companions the whole time until you added the males at the end, or if these females were kept virgins until you added the males at the very end. The result was the same, drastic reduction in the oocyte quality based on the readout that they can't support embryo development into larvae. And what's also very interesting, and it's very novel for her work, is that that flies have well-studied mechanisms of temperature sensing, and TRPA1 and GR28B are warm temperature sensors that have been very well studied in flies, but mostly in a behavior context, so that the flies can just avoid areas that are too warm to thermoregulate. But the question was, okay, these warm temperature sensors could potentially control more than just behavior. So Anna asked if they might be involved in controlling any of this response to oogenesis. And she found that for oocyte quality, TRPA1, which is highly evolutionary conserved, was required for this partially required for this reduced oocyte quality at 29 degrees. So if you compare the yellow-white control here, after 10 days at 29 degrees, it has a reduced oocyte quality. If you and we've seen this in other. We saw some of that in the last graph, but these are all at 29 degrees. But if the flies are homozygous mutant for TRPA1, this temperature sensor, the hatching rate and oocyte quality then are markedly improved. but not for the other temperature sensor, GR28B. But anytime you have TRPA1, homozygous mutant, the oocyte quality is improved relative to the wild type at 29 degrees. So then to summarize all of Anna's work, and I skipped a lot of the data because it's a lot of details to cover. So at cold temperature, the main mechanism for reducing egg production is a reduction in follicle growth. And at warm temperature, the main mechanisms are death of early germline cysts and of vitellogenic follicles. And in addition to this reduced egg production, there's a major effect of warm temperature in dropping oocyte quality, which is partially dependent on this conserved temperature sensor. And what I did not have to mention or show you the data for is that I did mention actually that the cold temperature has a positive effect on early germline cyst survival. They survive even better than flies at the controlled 25-degree temperatures. And what I did not have time to show you is that the cold temperature also has a very positive effect on germline stem cell maintenance. So instead of undergoing their gradual loss of stem cells at 25, the stem cell numbers remain strong until at least 20 days at 18 degrees. And similarly, incubating flies at 18 degrees also had a marked effect in improving oocyte quality, so that the oocyte quality remained even better than the 25-degree females of the same age. So finally, I would like to acknowledge the current and past members of my lab, and I mentioned a lot of them by name. And right now I have a small but potent lab, and you know Rodrigo and Ana. I mentioned Ana already, but Rodrigo is also in my lab, and he was also in your program, in your department, in your UFRJ. And Emily and Reham are two graduate students, and I'm blaming them for giving the talk in English because they said they were going to attend the seminar. And with that, and thanking all the very generous Drosophila community and my sources of funding, I'll be happy to take questions. Thank you.
Thank you, Daniela. It was a fantastic talk. I think it was amazing. So eu just talk a little bit em português. Pessoal, se vocês quiserem mandar as perguntas, vocês podem mandar no chat em português ou em inglês, que a gente traduz, assim, não tem problema. Tá? Podem ficar à vontade. Mas vou continuar falando em inglês nas perguntas, porque o pessoal que está assistindo. So back to English. I have some questions. I will start. I was looking at the data. I think you didn't show us, but you said that you make a screening of RNAi for all or most of all nuclear receptors to look for the oogenesis effects. Mm-hmm. And then about the 7-up nuclear receptor, then in that paper, you first you make like a general RNAi effect, then you go with specific for each organ using drivers. But I have a question about the fat body specifically. When you knock down 7-up in the fat body, do you see any effects on did you look for effects on the fat body, like for lipid metabolism or I was just thinking because we don't know anything about 7-up, so I was thinking maybe it is involved in lipid metabolism or something, or metabolism the fat body.
Actually, hang on just 1 second.
Okay.
I'm at home and my kids are not here. Just 1 second. Desculpa também a interrupção lá quando os meninos estavam me chamando.
Sem problemas. A gente está acostumado, isso é normal, acontece o tempo inteiro.
Um carteiro tentando entregar uma carta e acabou que não entregou. Não sei. Okay, the 7-Up, sorry. That's Zoom life. 7-Up, yeah. So when, a lot of nuclear receptors have major roles in controlling metabolism, right? It's actually hard to find one that doesn't have something to do with metabolism. But in the case of 7-Up, we thought it might have something to do with metabolism too. But when Leslie did the RNA-seq analysis, and that's also published, like I didn't have time to go through it, and she looked at all the transcriptome differences when you knock down 7-Up in the fat body. She was actually knocking it down both in the adipocytes and in the inocytes at the same time for the RNA-seq. There is no change in metabolic genes.
Okay.
So 7-Up is one of those rare nuclear receptors that has nothing to do with metabolism.
Okay. Nice to know.
Instead, we saw these other genes that are more related to immunity and detoxification responses and.
Oh, that's nice.
Yeah. So
actually, because I would expect something on metabolism. So that's
I know we did too, but it's not the case.
And in that screening, did you get any effect on the knockdown of HNF4 on oogenesis?
Yeah, she saw some effects, but she was knocking it down in somatic cells.
Mm-hmm.
Yeah. But we didn't publish the actual screen. We did the screen, but she only published the ones that she characterized in more detail.
Okay.
Because, but the reality is that, not surprisingly, pretty much all nuclear receptors have some sort of role in oogenesis or not.
Okay.
The screen, it was like almost everything affected oogenesis, and I was like, well, of course. The question is how.
Yeah, that's interesting because we were talking before the talk. A student of mine was on the United States last year, and then she made like overexpression of HNF4 in the fat body. And when we overexpress this nuclear receptor, then we see a reduction on the egg production. But we think that's because as the HNF4 is involved in lipid catabolism, then they draw up all the most of the lipids, and then there is less lipids for the cysts of eggs. But then we make a overexpression on some parts of the ovary, and then we saw an increase in the egg production. Although the quality of the egg is not so good, because we have more eggs but less larvae in adults showing up level. And actually we don't have we don't know why directly why this happened when you overexpression. So it would be nice to see the data on knockdown with HNF4 to see if there is something of a complementation, something like that.
Yeah, but she didn't. The initial screen that she did was just with like egg laying, and she only analyzed in depth the ones I mentioned, 7-Up and HR4. Yeah,
that would be nice to see.
Yeah, it would be nice to figure out what all these nuclear receptors do.
Yeah, there are a lot of them also.
But then there's like too many questions. There's other things we want to do too. So
I'm curious for nuclear receptors. Now, do you have questions? Because people in the chat is like not talking too much.
I hope that
your microphone is turned down. Naomi, microfone está desligado.
It's not a question, but it's a I was surprised to see the figure about which enzymes are on when the food is rich or food is poor. That figure is so rich to see, né? Enzymes working, not working. I think we can follow up to all the organisms. Can be.
Yeah, and actually that paper, of course, we have all the files with all the data in that paper, but we saw that kind of on-off enzymes, or not high-low enzymes, for a bunch of other different metabolic processes. So it's not like those are just because we were like, it's just too much data, right? And we wanted to publish the eye track, but we didn't want to just publish the data. So we followed up some of the exam, but there's a lot more to follow up, and Rodrigo will probably follow up some of that in Drosophila at some point. But there's a lot more, and there's actually other things. Like we found that there's rapid changes in lipofluorin that's involved in transporting lipids.
Oh, nice.
And that's
I like lipofluorin also.
Yeah, and we saw changes in ferritin that transport iron. And actually, if you knock down ferritin from the adipocytes, there is loss of germline stem cells. So that's showing like specific nutrients, right? And we also, and that's a different paper we published, we saw changes in extracellular matrix proteins, including collagen 4. And then we followed up. Leslie also did that work, and we have a paper in, now I can't remember, I don't remember what journal, but it's also Leslie Weaver, where she showed that collagen 4 is required in the adipocytes to maintain germline stem cells in the ovary, because we showed that the collagen 4 made in the adipocytes gets transported to this extracellular matrix around the niche, and there it affects the levels of icadirin and whether or not the stem cells are maintained. And that's only in the niche, because the later parts of the ovaries where the follicles are developing, they make their own extracellular matrix. So, and then it's not affected by the fat body. So that was kind of interesting too. And we see that not just for collagen, and it's just 12 hours. We see like a bunch of them increase levels at the same time. And it's probably because they are not getting secreted and transported away from the fat, right? So because we were looking at proteins, it could be changes in expression, could be changes in secretion, could be changes in degradation. So but we followed up some of them, but there's so much data in that paper. That we didn't, again, like, and that's what, there's too much open area for research in this field. So, and I just get interested in something else, or the person in my lab is interested in something else, and then it's not possible to keep following
to do everything.
Yeah.
So, I think we don't have questions for the people that was watching.
Eu espero que tenha dado para todo mundo me entender, apesar de...
Não, hoje foi excelente.
Foi. Eu fui entender sim, com certeza.
Eu fui entender com certeza.
Se não, vocês mandam e-mail para o Rodrigo e para a Ana pedindo para explicar.
Olha que o pessoal manda, hein?
Pode mandar.
Bom, pessoal, então, queria agradecer de novo a professora Daniela por ter aceitado o convite de falar aqui para a gente, foi sensacional. E na semana que vem a gente vai ter a professora Elisângela, que é da Faculdade de Farmácia, que vai falar sobre riscos relacionados ao uso de antimicrobianos, em pacientes pediátricos. E aí, semana que vem a gente está de volta. Obrigado, professora. Tchau, tchau.
Obrigada, Daniela.
Muito obrigada.
Tchau.